Hi guys, my name is Suraj Kadam and today we discuss on action of metformin. Metformin is used by diabetic patients. Metformin is widely used drug that results in clear benefits in relation to glucose metabolism and diabetes related complications. The mechanism underlying these benefits are complex and still not fully understood. Physiologically, metformin has been shown to reduce hepatic glucose production, yet not all of its effects can be explained by this mechanism, and there is increasing evidence of a key role for the gut. At the molecular level, the findings vary depending on the doses of metformin used and duration of treatment. With clear differences between acute and chronic administration, metformin has been shown to act via both AMP activated protein kinase and dependent and AMPK independent mechanisms by inhibition of mitochondrial respiration, but also perhaps by inhibition of mitochondrial glycophosphate dehydrogenase and a mechanism involving the lysosome. In the last 10 years, we have moved from a simple picture that metformin improves glycemia by acting on the liver via AMPK activation to a much more complex picture reflecting its multiple modes of action. More work is required to truly understand how this drug works in its target population. Individually used by type 2 diabetic patients. The drug metformin and related drug fenformin are derived from the galegine, a natural product from the plant Galega officinalis, used in herbal medicine in medieval Europe. Galegine was tested as glucose lowering agent in humans in the 1920s but was found to be too toxic. At about the same time, two synthetic derivatives of galegine, metformin and fenformin, were first synthesized and tested, although they were not introduced to clinic, clinical use until the 1950s. Chemically, galegine is an isopropyl derivative of guanidine, while metformin and fenformin are biguanides containing two coupled molecules of guanides with additional substitution. Unlike most modern drugs, metformin is therefore derived from a natural product used in herbal medicine and was not designed to target a particular pathway or disease mechanism. It was established as a safe and effective therapy before detailed mechanistic studies because possible, and despite its clinical use for 60 years, its molecular mechanism of action remain much debated. In this brief review, we summarize the current evidence highlighting how metformin's benefits are likely to be caused by a variety of molecular mechanisms. Oral dosing of immediate release metformin in humans, approximately 70% of the dose is absorbed from the small intestine, with the remainders passing into the colon before being excreted in feces. Metformin is excreted in urine unchanged with no metabolites reported. Plasma concentrations of metformin in humans are typically in the low micromolar range but are 30 to 300 times higher in jejunal samples. A recent metformin positron emission tomography study demonstrated that oral metformin becomes highly concentrated in the intestines, livers, kidneys, and bladder, with only slow accumulation in muscles. In this study, the hepatic tissues systemic blood activity was under 5 oral dosing of the metformin tracer, demonstrating that much greater metformin concentration are achieved in the liver than in the plasma. Extrapolating from systematic concentrations would estimate hepatic concentration post oral dose in rats dosed with metformin. Accumulation was observed in the pancreas and adipose tissues at a concentration of approximately half that seen in the liver. How? The human pharmacokinetic data point to the liver, kidney, and intestines as the key target organs of metformin, and in this review, we will primarily focus on the liver and intestines, particularly when referring to the beneficial impact of metformin on metabolism and inflammation. Other mechanisms relating to potential cardiovascular benefits, cancer prevention, and aging are covered elsewhere in this issue of diabetics. Next is metformin and the effect on liver. Metformin is traditionally thought to act on the liver to improve blood glucose levels. To several lines of evidence support this. First, in mice lacking the organic action transporter 1, which take up little or no metformin into the liver, metformin was ineffective at improving blood glucose after high fat feeding. Second, tracer studies in humans show that metformin lowers hepatic glucose production with minimal impact on peripheral insulin mediated glucose uptake. However, when only placebo controlled studies were analyzed, the impact of metformin on endogenous glucose production (EGP) was not significant unless concomitant drug induced reproduction reductions in plasma insulin were used to adjust EGP. Third, as will be summarized here, multiple studies in mouse hepatocytes, the transgenic mice provide evidence for a role of metformin in reducing hepatic gluconeogenesis or insulin sensitivity. The next point is metformin and the mitochondrial control of hepatic gluconeogenesis. The gluconeogenesis is an energy intensive process consuming 6 ATP equivalents per molecule of glucose synthesized. Hepatocytes need to balance the demand for ATP with supply, with the latter primarily provided by mitochondria. Metformin accumulates within mitochondria to concentrations up to 1000-fold higher than in the extracellular medium. Because metformin carries a positive charge and the membrane potentials across the plasma membrane and mitochondrial inner membrane drive metformin into the cell and subsequently into the mitochondria. The most intensively studied mitochondrial action of metformin is the inhibition of complex 1 of the respiratory chain, which suppresses ATP production. A persistent criticism of this mechanism has been the higher extracellular concentrations required to observe rapid effects, although lower concentrations of metformin do inhibit complex 1 in rat depetoma cells after several hours. This delay was ascribed to the slow uptake to metformin by mitochondria, which has recently been observed experimentally. In addition, some studies do not detect any changes in cellular ADP-ATP ratios after metformin treatment, although they can be observed with fenformin in cells carrying out gluconeogenesis. Concomitant suppression of this pathway might explain most effects on ADP ATP ratios. Other consequences of respiratory chain inhibition besides ATP production, such as changes in the NAD NADH ratio, may also contribute to the effects to metformin on gluconeogenesis. The center of metformin's mechanism of action is the alteration of the energy metabolism of the cell. Metformin exerts its prevailing glucose lowering effect by inhibiting hepatic gluconeogenesis and opposing the action of glucose. Metformin acutely lowers blood glucose levels by inhibition of intestinal glucose transport.